Can you taste it? A chemical called phenylthiocarbamide, or PTC, makes vegetables like cabbage and Brussels sprouts taste bitter. But some people can't taste it. Are your earlobes attached directly to your head, or are they free? Both earlobe attachment and the ability to taste PTC's bitterness are traits inherited in a pattern predicted in the 19th century by an Austrian monk named Gregor Mendel. In his monastery garden, Mendel performed breeding experiments with the garden pea that led him to derive laws of inheritance. While Mendel studied peas, the fundamental principles of inheritance he discovered apply to humans and other animals because the mechanisms are essentially the same for all complex life forms. Mendel's laws are derived from a carefully planned and controlled scientific process. In this presentation, we'll first look at Mendel's experiments. Then we'll see how his observations led to the laws of segregation and independent assortment. Why did Mendel choose the pea? First, peas have many easily distinguishable features. For example, pea flowers are either purple or white. Mendel studied 7 individual traits: flower color, flower position, seed color, seed shape, pod color, pod shape, and plant height. Second, peas are easy to grow, so Mendel could collect data from thousands of plants, making his statistical analysis more accurate. Finally, Mendel could manipulate their reproduction to control which plants mated with which. Pea plants usually fertilize themselves, as pollen released from the male reproductive organ, the stamen, lands on the female reproductive organ, the carpel. Mendel performed cross-pollination, or fertilization between plants, by removing the stamens from a plant and brushing pollen from the stamens of another plant onto the carpel with a paintbrush. Mendel used only true breeding plant strains in his experiments. A true breeding strain is composed of identical individuals that always produce offspring with identical characteristics when cross-pollinated. Before starting his experiments, Mendel cross-pollinated plants for several generations to make sure they were true breeding. An X between parents indicates a cross, and an arrow connects parents with their offspring. Mendel studied each of the 7 traits one at a time for many generations. Let's use his study of flower color to understand his experimental method. Mendel's first set of experiments involved a monohybrid cross, which is a cross between 2 individuals that differ in only one trait. For example, he crossed true breeding white-flowered plants with true breeding purple-flowered plants. The original generation of any crossing scheme is called the parental or P generation. The offspring of the parental cross belong to the first filial or F1 generation. Filial means relating to offspring. After he obtained the F1 plants, he allowed them to self-pollinate. The offspring of each F1 plant made up the F2 or second filial generation. To make sure his calculations were statistically accurate, Mendel counted large numbers of offspring in each generation. Now let's learn about Mendel's laws. Through his monohybrid cross experiments, Mendel made 4 key observations that led him to propose his first law of inheritance. First, Mendel noticed that each trait, such as flower color, has 2 alternative forms. For example, purple and white. He proposed that each trait is controlled by a different gene, and that every alternative is specified by a different form of that gene, called an allele. For flower color, Mendel assigned a capital P for the purple flower allele, and a lowercase p for the white flower allele. We'll discuss the use of capital versus lowercase letters in just a minute. For each trait, an organism inherits 2 alleles, one from each parent. An individual is homozygous if both alleles are identical, while a heterozygous individual has 2 different alleles. The allelic composition of a gene is called the genotype. The physically observable translation of a genotype is called the phenotype. For example, a pea plant with the genotype big P has the purple flowers phenotype, while a plant that's small p has white flowers. Generally speaking, true breeding strains are homozygous. Mendel noticed that none of the individual traits blended in the offspring. For example, each of the F1 and F2 plants had either purple or white flowers. There were no in-between colors. For each of the 7 traits studied, Mendel found that plants inherited either one or the other alternative. He never saw anything in between. From this observation, Mendel proposed that genes are distinct factors. In various ways, alleles remain distinct and are passed on to offspring unchanged. Mendel observed that traits that don't appear in an individual are still passed on to offspring. For example, he saw only purple-flowered plants in the F1 offspring, while the F2 generation had both purple and white-flowered plants. From this observation, Mendel proposed that there are 2 types of alleles, dominant and recessive. A dominant allele only needs to be present in one copy for the phenotype to be seen. For pea plants, the allele that determines purple flower color is dominant. A capital letter indicates a dominant allele. 2 copies of a recessive allele are needed to see the corresponding phenotype. For pea flower color, the white flower allele is recessive. A lowercase letter indicates a recessive allele. Purple flowers result from homozygous big P or heterozygous big P small p. Homozygous small p plants have white flowers. Since his parental plants were true breeding, Mendel reasoned they must be homozygous big P and small p. The purple-flowered F1 plants were heterozygous big P small p, and the purple-flowered plants in the F2 were either homozygous big P or heterozygous big P small p, while the white-flowered plants were homozygous small p. Mendel also observed dominant and recessive phenotypes for the other traits he studied. Finally, Mendel observed that the ratio of dominant to recessive phenotypes in the F2 generation was always the same. He consistently got 3 offspring with the dominant phenotype for every 1 offspring with the recessive phenotype. Mendel saw the 3-to-1 ratio for all 7 traits. From this observation, Mendel proposed that the 2 alleles for each gene segregate randomly into gametes. Recall that a gamete is a sperm or egg cell. He also proposed that gametes contain 1 allele of each gene and that they unite randomly. This makes sense to us because we understand meiosis. But Mendel didn't know about meiosis. From his 4 observations, Mendel proposed his first law, the law of segregation. Allele pairs separate during gamete formation and then randomly unite when gametes fuse during fertilization. A grid called a Punnett square shows how Mendel's monohybrid cross results are predicted by the law of segregation. A Punnett square is used to diagram the expected types and ratios of each potential offspring genotype of a cross. We write the possible gametes from one parent across the top and those of the other parent along the side. 1 allele goes in each box. It doesn't matter which parent goes on which side. We fill in the boxes by copying the alleles across or down into the empty squares. This gives us the predicted frequency of each possible genotype. 100% of the offspring will be heterozygous big P small p, so they'll have purple flowers. This is exactly what Mendel observed. Let's use a Punnett square for the F1 generation. Self-pollination of Mendel's F1 offspring is analogous to having 2 heterozygous big P small p parents. Test your mastery of Mendel's first law by filling in the progeny genotypes in the white boxes of the Punnett square. Click Submit when you're done. Click Jump Ahead to skip this step. We expect 1 out of 4 big P offspring, 2 out of 4 big P, and 1 out of 4 small p. This translates to 3 purple offspring for every 1 white offspring. This is exactly what Mendel saw. A Punnett square is one method of determining probability. To learn more about probability, see the reference sheet. When plants have the dominant phenotype, how do we know whether they're homozygous for the dominant allele or heterozygous? Mendel determined the genotypes of dominant pea plants by performing test crosses, which are matings between an individual of unknown genotype and a homozygous recessive individual to determine the unknown genotype. The offspring phenotype ratios from a test cross will be different if the individual being tested is homozygous or heterozygous. We'll use Punnett squares to compare the progeny phenotype ratios. If the tested plant is homozygous dominant, all offspring will be purple. If it's heterozygous, half of the offspring will be purple and half will be white. Mendel carefully observed inheritance patterns one trait at a time. Now let's learn what happened when he studied multiple traits at the same time. After formulating his law of segregation, Mendel studied inheritance patterns using the dihybrid cross, which is a cross between individuals that differ in two traits. For example, Mendel studied inheritance in plants that differed in both seed color and seed shape. Yellow, or big Y, is dominant to green, or small y, and round, or big R, is dominant to wrinkled, or small r. If a true breeding plant with yellow round seeds is crossed with one with green wrinkled seeds, Mendel reasoned that the big Y, big R gametes and the small y, small r gametes would make doubly heterozygous yellow round offspring. This is exactly what he found. Next, Mendel wondered if the big Y and big R alleles and the small y and small r alleles from the original parent plants would somehow stay together when the F1 plant formed its gametes, or if they'd mix and match to make gametes with new allele combinations. If the parental alleles somehow remained together, Mendel would have observed a 3-to-1 ratio of yellow round to green wrinkled offspring. Mendel didn't observe this ratio. Instead, all possible phenotype combinations occurred in a 9-to-3 to 3-to-1 ratio. Mendel reasoned that the seed color and seed shape alleles combined in many different ways to make gametes. Let's use a Punnett square to see how these four gamete types combine to give this offspring ratio. Yes, in fact, a 9 yellow round to 3 green round to 3 yellow wrinkled to 1 green wrinkled ratio is predicted by the genotypes in our Punnett square. Mendel observed similar 9-to-3 to 3-to-1 ratios with all of his dihybrid crosses. These results led to Mendel's second law, the law of independent assortment, which states that each allele pair segregates independently during gamete formation. This makes sense to us because we discussed independent assortment in the previous chapter about meiosis. Independent assortment is the random distribution of the maternal and paternal members of each homologous chromosome pair to the daughter cells. If we look at a cell that's heterozygous for 3 genes, A, B, and C, we see that according to the law of independent assortment, 8 different genotypes of gametes can be formed. Mendel's laws apply to all diploid organisms, including humans. For example, the PTC bitterness gene has alleles big T for taster and small t for non-taster. The taster allele is dominant, and the non-taster allele is recessive. A person can taste PTC if they are homozygous big T or heterozygous big T small t, while a homozygous small t person can't taste it. The earlobe attachment gene is also inherited in a Mendelian way. The big F allele is for free earlobes, and small f is for attached earlobes. Free earlobes are dominant, and attached earlobes are recessive. A person with free earlobes is either homozygous big F or heterozygous big F small f, while a homozygous small f person has attached earlobes. A homozygous PTC taster man with attached earlobes has big T little f gametes. A PTC non-taster woman homozygous for free earlobes has small t big F gametes. If they have a son, he'll be heterozygous for both PTC bitterness and earlobe attachment. Because the alleles of these two traits assort independently, the son can make 4 types of gametes, 2 of which are different from the parental gametes. If the son has children with a woman who's heterozygous for PTC bitterness with attached earlobes, their gametes can combine in 8 different ways to yield 4 possible offspring phenotypes. To learn about a statistical method for determining whether alleles always assort independently, see the reference sheet. We've just explored Mendel's laws of inheritance and learned many vocabulary terms. Alternative forms of a gene are called alleles, which can be dominant or recessive. A genotype is the allelic makeup of an organism. Organisms are heterozygous when they have 2 different alleles, and homozygous when they have identical alleles. A phenotype is the physical translation of a genotype. Mendel's experimental method was a carefully planned and carried out scientific process. He gathered data on thousands of plants. Mendel's law of segregation states that allele pairs separate during gamete formation and then randomly unite when gametes fuse during fertilization. His law of independent assortment states that each allele pair segregates independently during gamete formation. A Punnett square can be used to predict the types and ratios of offspring for monohybrid and dihybrid crosses. Mendel's laws of inheritance apply to all diploid organisms, including humans. Both PTC bitterness and earlobe attachment are inherited in a Mendelian fashion. Inheritance of some other human traits is more complex. But such patterns still depend on the fundamental genetic groundwork performed by Mendel.